Biochemically important monosaccharides. Of the many monosaccharides, 6 are particularly important in the functioning of the human body. These are the glyceraldehyde, dihydroxyacetone, glucose, galactose, fructose, and ribose. The first one is the D-glyceraldehyde and dihydroxyacetone. They are the simplest of the monosaccharides. As you can see, glyceraldehyde has a carbonyl group that is located at the terminal end. This is an example of an aldehyde. Dihydroxyacetone has a carbonyl group that is located at the center. This is an example of a ketone. They are important intermediates in the process of glycolysis. Next one is the D-glucose. As you can see in the picture, the glucose is an example of a sugar with an aldehyde group. It has 6 carbon atoms. Of all the monosaccharides, D-glucose is the most abundant in nature and the most important from a human nutritional standpoint. Ripe fruits, particularly ripe grapes, are good sources of glucose. It is often referred to as grape sugar. 2 other names for D-glucose is also given, and they are dextrose and blood sugar. The D-glucose tastes sweet. It is nutritious and it is an important component of the human diet. 2 hormones, namely the insulin and glucagon, are involved in glucose. Insulin, which lowers the blood sugar level, and glucagon, which increases the blood sugar level. They are important in keeping the glucose blood concentration within the normal range. Abnormal functioning of the hormonal control process for blood glucose level leads to a condition which is called diabetes. The next one is D-galactose. Galactose is one of the components of the disaccharide lactose. It is synthesized in the mammary glands in lactose, milk sugar. Sometimes it is known as a brain sugar. D-galactose is also an important marker in determining the blood type of the person. A comparison of the Fisher projection formula for D-glucose and D-galactose. As you can see, galactose is similar with glucose. They are both with carbonyl group that is located in the terminal end. It means to say they are aldehydes. They also have 6 carbon atoms with difference on the 4th carbon because for glucose, the hydroxyl group is attached to the right. For galactose, the hydroxyl group is attached to the left. D-galactose is sometimes encountered as a free monosaccharide. However, it is a component of numerous important biochemical substances. Again, as what we have mentioned, it is part of the milk production. It is part of lactose, which is an important disaccharide. D-galactose is also called brain sugar because it is a component of the glycoprotein that is found in the brain and nerve tissue. Patients with galactosemia lack an enzyme that is needed to metabolize galactose, which accumulates and causes cataracts in serum. The next sugar is D-fructose. It is one of the components of the disaccharide sucrose. It is also known as levulose and fruit sugar. Let us now take a look at the picture and see if there are similarities and differences between these 2 sugars. First, let us take a look at glucose. The carbonyl group is present in the terminal end. Therefore, our glucose is an aldehyde. Let us now count the number of atoms, specifically carbon atoms for glucose and fructose. Carbon has carbon number 1, 2, 3, 4, 5, and 6. Let us now count fructose. Fructose has 1, 2, 3, 4, 5, and 6. Therefore, both glucose and fructose contains 6 carbon atoms. The terminal end that is found on glucose is an aldehyde. What about for fructose? As we can see in the picture, the carbonyl group of our fructose is located on the second carbon. Therefore, this is a ketone. Next, let us take a look at carbon number 3 for glucose and fructose. The hydroxyl group that is attached to the third carbon is located in the left side. What about for fructose? Yes, it is also located in the left side. This is also the same for carbon number 4, wherein their hydroxyl group is attached to the right side. Same as true with fructose. Next, carbon number 5, the hydroxyl group is also located on the right side. This is also the same for fructose. And lastly, we have the last alcohol, so CH2OH. This is also the same for fructose. Aqueous solutions of naturally occurring D-fructose rotates the plain polarized light to the left, hence the name levulose. It is a ketohexose that is found in honey and almost twice as sweet as table sugar with the same number of calories per gram. Ribose is a component of a variety of complex molecules. This includes ribonucleic acid or RNA and energy-rich compounds such as ATP or the adenosine triphosphate. Let us take a look at the picture and let us try to differentiate ribose from deoxyribose. Let us count the number of atoms for each of them. Ribose contains 1, 2, 3, 4, and 5. What about deoxyribose? 1, 2, 3, 4, and 5. It means to say they both have 5 carbon atoms. Let us take a look at a very important carbon wherein we will see their differences. As you can see, the differences is on the second carbon. Okay? For ribose, the hydroxyl group is attached on the right side. Hydroxyl group is made up of oxygen and hydrogen. But what about deoxyribose? What is missing for the second carbon is the oxygen. So that is their main difference. On the second carbon, ribose has hydroxyl group, whereas deoxyribose, we do not have oxygen that is present on the second carbon. Now, let us take a look at the third, fourth, and fifth. So, as we have observed, the hydroxyl group are same. They are all located on the right side of the carbon atom. So, this is the D-ribose in relation to the deoxyribose. Their main difference is deoxyribose do not contain oxygen on the second carbon atom. So going back, as we have mentioned a while ago, your D-ribose is also important compound in the DNA, which is the 2-deoxy-D-ribose. This is an important part in nucleic acid chemistry.